Isolagen, a pioneering life sciences company, specializes in the research, development, and commercialization of autologous cellular therapies to treat the signs and effects of aging. The proprietary Isolagen process is currently available in the UK and is in Phase 3 clinical trials in the US. The process uses a person's own living cells to strengthen and rebuild the skin's natural foundation. Thousands are enjoying the benefits of the Isolagen process in the UK, where it's been well received by patients and medical professionals alike. Beyond dermal indications, Isolagen is researching the use of autologous cellular therapies to treat gum recession, acne scarring, and wound healing, among other indications. With its patented technology, Isolagen believes it is uniquely positioned to compete on a global scale, initially in the non-surgical cosmetic market, and then with broader market applications, bringing autologous cellular therapy to the rest of the world.
We are taking steps now to prepare for a future in which the Isolagen process will be available worldwide for a variety of cosmetic and medical indications. One of the most significant steps addresses the very heart of the Isolagen process, updating the way we grow the patient's cells. Over the past 2 years, we've invested significant resources to create an automated cellular expansion system, or ACE, to upgrade our manufacturing process to be scalable, consistent, and more efficient. Not only will the ACE system enable Isolagen to take on global markets efficiently, ACE will create an entirely new revenue stream through the licensing of the technology to the many cell and tissue growth companies worldwide, reinforcing our position as a global leader and pioneer.
In order to understand the true value of the ACE system, it is important to take a close look at the current manufacturing methods employed by Isolagen. The current method begins in the physician's office, where a very small sample of tissue is taken from the area behind the patient's ear. The tissue sample, which contains collagen-producing fibroblast cells, is then labeled with the patient's personal tracking number, packed and shipped overnight to the Isolagen laboratory, where the cell expansion process occurs. Once in the lab, fibroblast cells are separated from the patient's tissue sample and transferred into different-sized flasks as they propagate. This process yields only enough cells for one injection. Upon harvesting this injection, the process must be repeated multiple times to produce the correct dosage for a complete Isolagen treatment. Each individual treatment vial is then packaged and shipped to the physician for injection, approximately 14 days apart. Cell propagation in the current expansion process is effective but labor-intensive. Several years ago, a team of Isolagen scientists and engineers set out to design an automated cellular expansion system, ACE, that would be commercially scalable and efficient. As a result of their work, Isolagen believes this design of the ACE system is an elegant, simple, and complete solution for the delivery of the Isolagen process to the patient, and will revolutionize the way Isolagen and other tissue engineering companies propagate cells. Enhancements in the Isolagen ACE system design include securing the identification of each tissue sample from biopsy through injection by creating an electronic chain of custody linked to a specific patient biometric taken at the time of biopsy. Providing the first fully automated closed-loop cell propagation system made possible with the Isolagen ACE unit. Leveraging cryogenic preservation and freezing technology to allow maximum distribution and scheduling flexibility. The heart of the system as designed is the Isolagen ACE unit, which consists of two separate and distinct parts. The first is the permanent unit, which houses the circuitry and pumps that control the movement of fluids, and the thermoelectric heater, which provides heating and cooling without the use of incubators and refrigerators. The second part is the disposable unit, which contains the cell tower, media bags, cell separator, and all of the sterile tubing for media flow. This design eliminates nearly all human intervention, reducing the chance of contamination. Cells are initiated and then seeded within the cell tower, which is designed to accommodate the propagation of the cells on 9,000 square centimeters of surface area. This eliminates the time-consuming transfer of growing cells into new flasks, thus dramatically reducing the skilled labor and supervision needed in the laboratory. This design also decreases the amount of monitoring and quality control method testing needed. The Isolagen ACE unit, as designed, has a very small footprint, which allows for modular stacking and maximizes space efficiency. Each cell pod will contain 300 Isolagen ACE units and occupy approximately 80 square feet. And every step of this improved process, from feeding cells to the harvesting of injection syringes, will be executed in a sterile, closed-loop system. The benefits will be significant.
Our work in developing the ACE system demonstrates that we not only respond to market needs, we create market opportunities, increasing both our revenue and our competitive advantage, helping our company grow and rewarding our shareholders for believing in our shared vision. Being pioneers in the life sciences using the Isolagen process has and will continue to make a significant difference in people's lives around the world.
Isolagen, the science of living cells.